I will turn it over now to Abby Wakely, the Director of Communications with SECOORA, which stands for the Southeast Coastal Ocean Observing Regional Association. I hope that got it.
Thanks. Yeah, thanks so much, Brittany. You definitely got it. It is a mouthful like your guys' name. We love our acronyms here. I just wanted to quickly go over the agenda. Rachel's going to kick us off with an overview of the Sablo Islanders, and then we're going to hop into the SECOORA Data System with Axiom Data Science. And then we'll go into a data activity. And if you guys want, you guys can pull up your computer and look at our data portal at the same time. But this will be recorded, and it is something that you can recreate because we will send the recording to everyone registered. And as Brittany said, we have a great lineup of speakers. We've already heard from Brittany Dodge here, and then we have Rachel Guy from the Sablo Island National Estuarine Research Reserve, Lauren Showalter from Axiom Data Science, and Brian Stone from Axiom Data Science as well. So I'm going to stop sharing my screen, and Rachel, I think you can kick us off.
All right, hopefully you're looking at a PowerPoint slide that is not the other one. All right. Well, thanks for everyone for coming today. I'm pretty excited to talk to you about our monitoring program that we have through the NEARS network. So I'm going to be talking about the NEARS system-wide monitoring program. And one of the things I'm going to start with is kind of defining what a NEARS is, or what the NEARS is. So NEARS stands for National Estuarine Research Reserve System. And that system is really important to emphasize that we are, while each of the reserves are in place, we are part of a network that works together. And most people, when I ask them what is a NERS or have they heard of one, it's not one of the better known programs that are out there. So I'm going to go over briefly what this is. So what you see on the map here is all of the reserves that we have. And what we are is a network of 30 coastal sites that are dedicated to protect and study estuarine systems. The first one, which was Southern Slough in Oregon, was established in 1972. Saplo Islands is actually the second oldest, was established in 1976. Our most recent one, which was established in, I think in 2021, is in Connecticut, which leaves one last coastal state, which is Louisiana. And I believe that one currently is in discussion right now. So again, reiterating that this is a network of reserve systems that are all kind of dedicated to the same thing. Every reserve is a partnership. So, and it's a partnership between NOAA and between just a moment, I need to adjust how I am seeing things. There we go, that's better. Between NOAA and a state partner. And the state partner has to be a land management partner of some sort, such as a university system or a land management agency like a Department of Natural Resources. NOAA provides our guidance, and so all the reserves gets the same guidance from NOAA on what we're supposed to be doing and what we're supposed to be focusing on and they also provide the majority of our funding. However, the state partner conducts all the day-to-day operations and the site management as well. And it's important to note too that while NOAA gives us our marching orders, we still are site-based and we're place-based. So a lot of the things that we are doing while they were under NOAA's umbrella and mission, they are focused on what's happening within our particular sites. So Sablo Island is the National Estuarine Research Reserve. Again, we do love our acronyms. It's SINER for short. So if you hear me say SINER, that's what I'm talking about. Is a partnership between the Georgia Department of Natural Resources and Wildlife Resources Division and NOAA. And on the right, you can kind of see where Sablo Island is situated in the center of the Georgia coast, more or less. The yellow outline is our boundary. And while we don't encompass the entire island, this is on the west side, we have Little Sapelo. This is the Duplin River that comes up through it. And on the east side is the rest of Sapelo. While our reserve boundaries are not over the entire island, we do work with other island partners and do work within, throughout the island, not just within our reserve boundaries. So just reiterating our network, every reserve within the system has 4 core programs, and they all have the same missions within the programs. That's stewardship, training, education, and research. And I will touch on these briefly. Stewardship is focused on keeping the estuary that they're located in healthy. So they do this through monitoring, through vegetation mapping. So this habitat map was created with 2013 vegetation data. And then they also do restoration projects as well. Training program, which Brittany Dodge is our training program coordinator, looks at equipping locals and professionals, and as well as agency officials and local and state officials with the tools and knowledge that they need to be better stewards of the estuaries. So that could be in forms of, classroom workshops off island or on island workshops, actually getting in the field and being trained. I think this was a master naturalist class that Brittany was involved doing, or teaching the public, how to build their own rain barrel systems. So it really spans quite a range of activities. And then the education program, which focuses on teaching K-12 students, as well as their teachers and the public about the estuaries and conservation. One of the great programs they have, which is called TOTES, or Teaching on the Estuary, is a program for our K-12 teachers, especially our science teachers, and gives them the tools to take back to their students so they can teach about the estuaries within their own classrooms. And that also is a nationwide program. So every reserve is doing this. So last but not least, the research program, which is the one that I am in charge of coordinating on the island. And again, this is place-based or reserve-based research. So what's relevant to each reserve, and as well as monitoring to support the conservation and management of our estuaries and coastal ecosystems. And really what I'm going to focus on for the rest of this talk mostly is the monitoring programs that we have, in particular the system-wide monitoring program. And yes, it has its own acronym, SWMP, which is pronounced SWMP, which I'll be referring to a lot more because the rest of it's too much of a mouthful. So the purpose of the system-wide monitoring program is collecting site-based monitoring data to provide standardized quantitative measures to determine how conditions are changing in the short and the long term. And this has 3 basic components nationwide. One is collecting abiotic indicators of water quality and weather. 2, biological monitoring, mostly in the form of vegetation monitoring. And then watershed, habitat, and land use monitoring through creation of habitat maps. I am mostly only going to focus on the first one because this is probably the biggest and longest running monitoring program within the NERS system. If you have questions about the other 2, I'd be happy to talk with you about them later or in the Q&A. So I will start just illustrating using Sapelo as our illustration for the abiotic monitoring that we do through the SWMP program. Again, you can see Sablo Island on the right of the screen here, and I've highlighted 4 different sites. Essentially, it's actually 5 different sites there. One's kind of hiding behind the other of where our swamp sites are. So the first one I wanted to go through was our weather station or our meteorological station. And I'll take a moment just to go over this naming convention we have at the bottom, because this is going to be pretty important in the long term when you're looking for our data. So every reserve out there has a 3-letter code that designates that reserve. So Sapelo's is SAP. And then each swamp site within a reserve has a 2-letter code. And so Marsh Landing, what you're seeing here, which is our weather station, has the 2-letter code of ML. And then the last 3 letters are telling you what type of data this is. So this is met data or meteorological data. And this naming convention is true for all of our reserves, though each one will have different site names. So what you see on the left is our meteorological tower. It has been there since, I believe, 1999 or 2002. We have data since 2002 here that's available, and I'll go over more of that. And Marsh Landing, if you're not familiar with Sapelo, is actually on right as you get off the passenger ferry to get on the island. So if you've been to the island, you've certainly seen our Met Tower. So for all of the reserves, we use standardized sensors and standardized calibration schedules. So you're seeing here 2 of the types of sensors that we're using. And so you know that when you can compare this, that when you go, that if you want to compare the weather data between different sites, that you know that we're all using the same sensors and we are conforming to a SOP that is having us calibrate on a 2-year cycle for most of our instruments. Some of them are on a 1-year cycle. And all of these instruments are instruments that conform with the National Weather Service recommendation as well. And we have standardized data collection too. Every reserve's weather station is a near real-time station. And I'll explain what that means in a moment. Each station is logging data in 15-minute intervals, and it's near real time because it only uploads to the satellite once an hour. However, we go out physically and download data monthly from that and upload it to our central data management office. And I will go over where that data is later in this presentation. And the parameters that we are all collecting in the exact same way throughout the network is temperature, relative humidity, barometric pressure, wind speed, max wind speed, wind direction, photosynthetic active radiation or PAR, and total precipitation. There's some optional parameters that you might see other reserves doing, but these are the core ones that everyone must do. Similarly, we have our water quality stations on Sapelo, and every reserve must have 4, and at least one of them must be near real time. Some reserves are going to have more water quality stations than 4, but everyone at least must have 4. And again, the naming convention, as you can see, we have, the reserve abbreviation, the site abbreviation, and then WQ for the type of data that's being collected there. And this is our new swamp manager, Ivana. She is putting one of our sensors into a deployment tube at one of our swamp sites. This is Hunt Dock. And again, 1 for every reserve is going to be a near real time, so you can look at that data within an hour or so of when it was collected. And again, we're using standardized sensors. All of the reserves are using YSI data sondes. We have transitioned to XO2. There's an older series, the 6 series, that was used before the XO2, but YSI is no longer making the sensors for it. So those will all be phased out if they haven't been already. And we all have a regimented calibration schedule for these as well. And I'll go over a little bit for that. So we have standardized deployment and data collection. None of our sondes can be out in deployed for longer than 45 days. And generally, especially in the growing season when it's hot and we get a lot of biofouling or can get a lot of biofouling, we'll actually swap them out every 2 to 3 weeks instead. And so there's never really going to be an interruption in the data, because when we go and deploy, we install a freshly calibrated sonde into the deployment tube. On the left, you can see the standard deployment tube that all of the reserves are using as well. That's the YSI recommended schematic for creating these tubes. It's logging data in 15-minute intervals, and we upload them pretty much the day that we retrieve them. So they're getting uploaded at least monthly, sometimes more than once a month. And going into the central data management office. The parameters that we're measuring are water temperature, conductivity, salinity, turbidity, pH, dissolved oxygen, and water depth or level. And there are some optional parameters such as chlorophyll A that some reserves are doing, but these are the core mandatory ones that everyone must do. And then finally, we have our water nutrients that we are taking at each of our 4 sites. And again, you can see the naming convention. The only thing that's changed is the type at the end. Every site must collect at 4 sites minimum, and that is a monthly grab sample, all done within the same time frame. So you have to do all of your sites. I think they give you 48 hours to reach all of them, but you're supposed to get them all done at the same time. And during that time, you're supposed to be doing a 24-hour diel sample as well. And we're using standardized collection techniques for this too. So again, I can show you where the SOPs for these techniques are. So we're using a grab sampler. So we're sampling close to the level of where our sondes are actually measuring. And then we use a automatic sampler that samples in series, such as this ISCO sampler that's on the bottom there for our diel sample. We have to collect our grab samples within 3 hours of low tide. And the diel sample should be taken over 24 hours, every 2 hours at the minimum of 11 samples taken to capture a full tidal cycle. And so generally what we're doing is we're setting up the diel sample the day before we go and collect all of our grab samples. And the upload schedule of this is a little different, and I'll explain why. It's at least annually, but often happens quarterly. The parameters that we're looking at is orthophosphate, ammonium, nitrate, nitrite, nitrate plus nitrate, nitrite plus nitrate, and chlorophyll A. And we filter those in the lab, but we send those off to be analyzed by a NIRS approved laboratory that can do the analysis. So some NIRS do have that ability to do at their own site. We do not. So we send ours to Virginia Institute of Marine Science. And so the turnaround of that can sometimes be a delay of a couple months, but once we get it, it gets uploaded into the Central Data Management Office. And I've been talking so much about the Central Data Management Office. I want to go over what it is a little bit and where you can access it, and kind of the quality control we do for this data that is dictated by the Central Data Management Office or the CDMO. This QR code, if you've got your phone out, it will take you to that. I will try to put in the chat, I can do that right now, the link if anyone wants to visit it. Whoops. And so what you're seeing here is the home page. Nope, you're not seeing the home page. There we go. The home page of the CDMO. And I'll go through a little bit more of this. I wanted to walk through our quality control first. So as I said, we have a data upload schedule with the meteorological data. It is monthly. With our water quality, it's at least monthly. And then our nutrients, it's at least annual. So, what happens when we upload our data to the CDMO, so we have an FTP site, it goes through a QA/QC. And I want to be really clear that we don't change data and we don't delete data from our data set. We flag the data and we leave it up to the user to decide what should be removed and what should be from their analysis. We're not making that call for you, but we do flag it to help it be a lot easier to filter things out. So we absolutely do not delete data. There might be some corrections that are done, rarely, but it is with the CDMO oversight way down in the end of the review process. So when we upload our data, there is an algorithm with the CDMO that immediately spits back a file to the reserve that flags any data where it's reading outside the censor's limits. So if we're reading ridiculous, pH of like 18, I mean, that doesn't make any sense for where we are. That will likely be flagged. Or if data is missing, if a sensor failed, this is not going to necessarily catch sensor drift or other things like, hey, we were doing maintenance, or something was weird, or biofouling might have affected it. I've had brushes that we have a central brush that wipes off all of our sensors every 15 minutes to keep the sensor from fouling too much. And I've had those sometimes fall off, and then by the time you collect it, all of your sensors are covered with algae, and that's going to affect your readings. But that's why we go through that flagging with the reserve staff does. And they change all those initial flaggings to marks, flag codes that should be accepted, suspect, or rejected. And in addition to that, they create metadata so they can keep track of what has happened, when that biofouling event might have happened, when that maintenance have happened, when a sensor might have failed. And so there will be the flagging as well as the metadata, and all of that is resubmitted to the CDMO annually, and that becomes provisional plus data. It goes through one final check with the CDMO, and where they review all of what the metadata and the provisional plus data and look for any discrepancies or anything that the reserve might have missed. And once that data is done, it is considered authenticated data. However, you can go on and find all of this data, provisional, provisional plus, and authenticated right now on the CDMO. And I'm actually going to stop sharing this PowerPoint presentation and go over to the CDMO, if I can. Let's see. Not seeing it. All right, I know that's not the CDMO. Hang on a moment.
Hey, Rachel, and while you're pulling that up, as a reminder, if you all have any questions, please go ahead and raise your hand or type them in the chat box.
All right. Here we are at the CDMO website, and to get data, first I want to walk through what's on the home screen here. There's a couple ways you can view and download the data. You can click on this map to the left. You can click on whatever reserve you want. There is abbreviations of the real-time monitoring data here. So again, here is Sapelo's Met Station up here, but all the other San Francisco Bays, all the other ones are here too. So you can see what things are looking like right now at Sapelo, but there's more in-depth ways of doing this. So this is, if you click on the get data up here, there's 3 different ways you can look at things. I'll go through the data graphing system here if you just want a quick view of stuff. So I'm going to select Sapelo. I'm going to select the Met station here. Click down here. And it's going to give you our current conditions right away. But maybe I want to look at how things looked during, well, it was Tropical Storm Ian when it came past us. So that would have been the 28th. And then maybe I want to see how it looked for South Carolina, and it hit a little bit later there. So I'll do that to the 30th. I want to look at their Met station, and we're going to look at wind speed together. You can graph that. And it pulls it up. And so if you look at the top, the blue line, and this axis here is the Sapelo axis, and the green line is the North Inlet Winyah Bay axis over here. And you can see we peaked here on the 29th at about, this is about 36 miles per hour. And about 24 hours later, it was the North Inlet Winyah Bay had about 39 miles per hour. So you can look at things like this. I also found it interesting to look at Hobos Bay, which is in Puerto Rico. They were hit by Hurricane Fiona, if you recall. And what was really interesting about this one, and devastating for them, is that it wasn't actually a big wind event for them, but it didn't matter. Their wind speeds were, we were pretty comparable in their wind speeds. This is Hurricane Fiona right here, and it it's not the wind speeds for them, but it still affected them pretty significantly, as you know. And you can also change these graphs to look at it in English units if you're sharing it with the public, just to have it to be a little bit more readily translatable for them. And you can download the data here as well if you want, or get yearly files. But then there's another way to do that. So let's say you don't want to look at all the graphs. You can do the advanced query system, and you can do it through zip downloads, where it'll just give you an entire year at a time for each site, or you can do a custom query if you want to look at specific time frames. So maybe not throughout an entire year or something like that. Or you can merge the data that combines weather and nutrient data. And I just want to walk through this really quickly so you can kind of see what it's going to look like when you look for stuff. So I'm going to leave these all unchecked. So if you go down to Sablo Island here, you can see our water quality, our nutrients, and our Met data. And what you'll notice is there's a lot more than 4 here, and that's because we've got some sites that are no longer active. So this Barn Creek here, this BC is no longer active, Flume Dock is no longer active. And just to be extra confusing, we do have an active Lower Duplin water quality site, but we have a deactivated Marsh Landing water quality site. So when you're selecting your data, that's how you read these. And so if you want the ones that are still active, just keep that in mind, or check with the reserve you're looking at. So I'll submit all locations and proceed to the next step. You say the years that you are interested in and get files. I'm not going to complete this here. You would fill out your name here and all the fields, and then you hit submit. And what will happen, depending on how much data you want, is you might get an email within a few minutes, within a couple hours. If it's a lot of data, it might take a couple days. That will give you a link to your zip files that you can download. So that's how you get the data. And then last but not least, I wanted to talk a little bit about how you, about the data about the data. So if you want to know more about how we collected it, all those SOPs that I talked to you about, our manuals, you can get here, and you'd select which ones you're interested in seeing, and you can see our process for doing it. Same thing as getting the data, and they will email you links to those manuals. We have where you can look at our QA/QC procedures here. It talks about the statuses and what you'll see, and then some about the flags as well. This is done, and you can see this in much more detail in our CDMO data management manual. So if you want to read all about it, you can read about it there. And then how you cite our data here. So it gives you an idea how you cite our data, and then the use of our data. Kind of boilerplate, but you know, we don't take any liability for how you decide to analyze this data. We have gone through, if you decide to use data before it's QA/QC, that is on you, not us. And so, but you are permitted to use this data. It is public data, it's in the public realm. We do require that you cite the SWMP system and the reserve that you collected the data from in any publication that you are doing. And then also this is talks about the metadata that we have. And so when you download any of our data, you will automatically get the most updated metadata for that data. So I am going to go back briefly to my presentation. And I've gone over that. And the last thing I wanted to go over, because I always get questions about this, about how do you come out and do research on Sapelo Island? And the first thing I want to tell you is that it's a limited access island, which means you can't just come on the island. You have to be a guest of someone who either resides on the island or is one of the local island partners, such as UGAMI or University of Georgia Marine Institute. The LTER is out there, Signer, or the DNR, and just to get on the ferry. So you can't just walk up and get on the ferry whenever you want. So the first step you want to do is contact me, and you want to contact me many months in advance. And that is because we are resource limited. We are limited in housing, and we are limited in transportation on the island. And so just make sure you're thinking about this well in advance, especially if you're doing grants. Don't surprise me and tell me you've written me into your grant and want to start in a month. That's not going to work out very well for any of us. So having in mind what your research question is, what you think the duration is, the rough methodology, and then I can understand better what kind of support you may need. Are you going to need on-island housing? For how long? For how, how long are you going to be out here at each stay? Do you need long-term? Do you just need short-term stay? Are you going to need permits to be out here? Because if you're doing anything in our marsh, you still need to get the zone permits to work out here. And then once we've talked about it, I would highly recommend, if you haven't been out here, you arrange a site visit to come out to the island. That way you can really get a better understanding of the accessibility of your sites that you think you might want to use, and then just the logistics of getting to the island. Our ferry is a passenger only ferry, so you can't bring your vehicle over here. If you're going to be planning on being here for an entire year, we can discuss barging a vehicle over, but that takes some extra steps to get that done. And then finally, once we've kind of talked, you submit a research registration web form, and that QR code will take you there. And it's also on the Sapelo Island's website too. And there you want to upload your abstract of detailed methodology, the permits that you've received. You need to identify the duration of it and upload any waypoints of where your research sites are going to be. And so with that, that is the end of my presentation. So I'd be happy to take any questions if there are any.
Doesn't look like we have any in the chat right now. If anyone wants to raise their hand, we can do that, or we can go ahead and move on with Abby's presentation, and then we can come circle back. If there aren't any immediate questions for Rachel.
All right. Well, thank you, Rachel. And so I'm actually going to pass it off to the Axiom Data Science team. Axiom Data Science is the data manager for SECOORA. So all those great data visualizations you see on our website and the great links that I shared to portal.secoora.org, they are the ones that handle all that, and they are the experts. So Lauren and Brian, I'll let you guys take it away.
Okay, hi, my name is Brian Stone. I'm a software engineer at Axiom Data Science, and I was just going to walk through a quick demo of the SECOORA data portal. And you should feel free to follow along at home. I'll paste the link in the chat. And if you have any questions, please let me know. I'm not a scientist, so if you see something going on that you're more interested in, you know, raise your hand or whatever, or interrupt. I'm happy to go along with whatever. Okay, I'm gonna try to share my screen. Okay, are we seeing the SECOORA data portal or my emails? Okay, great. That's what I was hoping. Okay, so this is the homepage. You can get back to the homepage from anywhere in the portal by clicking on this home button or the little home icon on the left side. If you click on the logo at the top, that's going to take you off to the main SECOORA website. This little green bar is going to be on every single page. So it's your main navigation. Depending on how big your screen is, you might not see the words next to the icons, but you can always hover over each item to see what it does. Going down the home page, there's some quick links into search and the featured data set, which is gliders right now, or to explore real-time data. And we'll look at each of those things. A little further down, you can get release notes, documentation, and then more links into the portal. And then further down, we have these data views. We'll get more into these later, and I'll show you guys how to make them. But data views are a way to put together different data sets of all different kinds, a little bit like what Rachel showed on the NERS site. And then data categories takes you into some pre-selected search categories. So going back up to the top, I'm just going to work my way across the top here. So we'll click on catalog. So this is the main search interface. And from here, you can enter anything you want in the search bar. We might not have anything, but if you start to type something, for instance, Florida, you'll start to see some, suggestions for how to auto-complete that. And then you can click on one of those and, it'll search and so on. Further down the page, you can click on different search result types. So right now we're looking at all result types. And so there's a, data layers, sensor stations, gliders, all mixed together. But if you're just interested in a particular data type like sensor stations, then you can click on sensor stations. So from here, you get a couple new filters. So on the right side, you get tags, variable types, and affiliates. So for instance, if we were interested in oxygen data, I could click on oxygen saturation. So now I get 85 stations that are available to me. If I'm just interested in NERS stations, I could find the NERS affiliate and click on that. And so this way this is just a way to quickly zone in on what I'm looking for. Each of these stations I can click on to get data plots and more information about the station. So at the top, you can see the this is the source where the data came from. You can click on that to get all stations from that source. On the right side, we get data plots. We'll go a little more into data plots in a minute, but it's pretty easy to work with these. You can expand the search bar or the time slider. You can click on source to get to go back to where the source data was pulled from. And then we can click on the sensor itself and get a full page. This page is going to have the most information about this sensor data. If we or a partner has done any quality control, you'll see that here, and you'll see information about the quality control. I'm just going to go back to that station page. Most of the components of this chart you'll recognize throughout the application, and we're going to go into a different way to get at sensor data in a minute, but you should see most of the same things as far as accessing data. If I were to expand out the data, I can use this little expander button to just go to the full data set. I can zoom in by dragging, and you can see that the little time slider updated for me, or I can manually drag it, or I can click on the calendar and select dates. So this would be a way to, if I was interested in a hurricane or something, I could see what happened on the 28th. Once you've zoomed in on data that you're interested in, you can click on downloads. This will take you for sensor stations straight into ERDAP, which is an application for downloading and visualizing data. So you can click on dataset, charts, or metadata, or you can just download the data. So if I click on this download button, I am going to get just the time period that I'm looking at on the chart. So it'll get me, it's just a quick way to download a CSV or a .NET-CDF file. I can download an image of the chart that I'm looking at, or I can queue this for download. This is a tool for putting together a collection of downloads that I want to, save for later. I'm going to head back out to search and sort of make my way through the other search types. So I'll click on catalog again. So we looked at sensor stations. Next one is data layers. These are layers that are mappable using the mapping application. So you can see the map button at the top here. So from here, I can learn about metadata for each of the data layers. I can get downloads for each of the data layers, and I can add them to the mapping application. So a quick way to handle this is to just click on tags. So if I know that I'm interested in shipping and navigation, I can click on that, and so on. And you can click on multiples of these. So if I click on shipping and navigation and marine species, then I start to get a smaller subset of layers. Moving platforms, this is where we have gliders and cruises. And if there were animal telemetry data, we would have that here as well. Again, there are variable types on the right side, as well as tags. So again, I can quickly search. I can add these to the main map, or I can visualize the data. So I'll click on this glider. This was deployed fairly recently, I think. In the upper left, I get a map of where the glider went. On the right, I'm seeing a curtain plot, sort of a cross section with time as the x-axis. And then beneath that, I get a 3D plot of where the glider went. So let's see. I'm going to see if I can show you guys everything at once. So I can set this auto rotating so you can kind of see different parts of it. But as I hover over the curtain plot along the top, I don't know if you can see, there's a red bar that's sort of zooming across the 3D plot, and then a red dot on the map. You can also just get a profile by looking at the hover. You can do the same thing by hovering over the map. So again, I get a red bar on the right side to indicate where in the plot I am, and a red bar on the 3D plot. Downloads are available at the top here, all different formats. I can choose my color palette, I can interpolate, I can visualize all the different variables, so I can look at chlorophyll or whatever. Again, if there are any questions, please let me know. There are a couple other different kinds of data types, variable types. This is a way to see a compilation of sensor stations for each of these variables. So if you knew you were interested in pH, you could search for pH here and get all stations that have pH. In fact, I'm going to demonstrate that really quick because I wanted to show you. So this gives you a map of where the data is. You'll recognize these little hexagonal this little hexagonal heat map. We use this throughout the site as a way to summarize data. As you zoom in, you'll get actual dots that show where actual sensor stations are. So at this point, I can see actual sensor stations. I can hover over them to see what they are, and I can get a little preview of the data. On the right side, you're getting sort of a manifest of the stations available that have pH, and then this inventory. And this can be a nice way if you know you're interested in data from a particular time period. You can, figure out which stations have data during that time period. You can see a few of these stations have green bars. So those are stations that do offer quality control. So you'll end up seeing green, orange, and red bars, and that will indicate data that's good or bad. For stations that don't offer quality control, we have just the gray bars to indicate that there is data there. Affiliates, let me go back out to search, clear out my search term. Affiliates works the same way. So this is very similar to variables, variable types. Click on an affiliate and you can see all sensor stations from that affiliate, and then you can see a manifest of when data is available. So I'll demonstrate that really quick. The inventory page offers also a variable dropdown. So I can see, now we're looking at chlorophyll for Everglades National Park data, but we could instead look at oxygen, etc. So that's search in a very small nutshell. Back to the homepage. Let's head into mapping. So the two ways to get to the mapping portal are to click on map, or you can just click on explore real-time data. And this will jump you into the mapping portal with our real-time data layer loaded. If you're visiting the portal for the first time, you should see the real-time data layer when you go to map either way. So this is a collection of real-time sensors. Again, we're seeing a heat map. Right now it's showing air temperature. I'm just going to work my way down the legend on the right side to demonstrate what's there. This variable type dropdown, I can choose a different variable type. So for instance, if I wanted to see atmospheric pressure, I could click on the atmospheric pressure button, and now I'm seeing a heat map of real-time atmospheric pressure conditions. I'm just going to go back to, actually we'll leave it the way it is. Sources lets me filter on source. So for instance, if I'm just interested in NERS stations, I can scroll down. You probably can't see the drop down, but I can scroll down to NERS, and now I'm seeing just stations where the source is NERS. I'm gonna go back to all. I can also filter on platforms, so I could filter down to buoys and fixed stations, and then tags. So there's one tag, tier C, which refers to water quality tags. I can also just do a freeform search here. So I think Rachel said the, so if I start to type in SAP, then I get the Sapelo station. So this hex contains 5 stations right now. As I zoom in, I can see the 5 stations. And again, I can click on the station and bring up data. So this is the same chart that we were looking at before. The main difference here is most of the functions are under this little gear dropdown because there isn't space for them. But I can click on this and change my time binning or my auto scaling. I can also just make it bigger. So this is a good way to work with data, if you don't necessarily need to bookmark it, but you can explore the data. The piece that I didn't go over on the sensor station was time binning. We automatically bin the data according to sort of what the computer thinks is going to do best. But if you don't like the binning, you can change it. If you try to get too fine a scale, it's not going to let you. So you can't look at all data for 30 years, for instance. You can't look at all hourly data because that would sort of bring down your computer. But you can download all hourly data if there's a sensor station that has that.
Thanks, Brian. Hey, we did have a question from a student. He's finding it a bit difficult to get the best satellite data that covered the Florida Keys shoreline changes as a result of Hurricane Ian. And he's wondering if you all have satellite data on the catalog. Or if you can recommend a
real-time satellite data?
It's sounding like it through the question.
We don't, but I can show you how to search for it. Okay. So you can head back out to the catalog, or you can just, so I'm clearing out my search filters here, or you can go under find data And so you can see I've already searched for biodiversity, but I can search for whatever I want here. So if I were to search for satellite, I get satellite data products, but I don't think we're going to have, I think you're looking for like a visual product that's sort of taking pictures of the shoreline, and we don't currently have that. But there is this other satellite data. The other thing you can do here is you can say we are interested in data just off the coast of Georgia. I can zoom in on an area and then click on spatial. And so this is now going to filter to data that is within the bounding box of where my browser is. And I can search on that. I can look at layer types. So right now, the only result types that we offer in the map search are data layers and platforms. Or I can bring up that tags interface, and I can look for just real-time data. So that would be another place to look for if you were looking for something like real-time satellite data. Click on the real-time tag and see what's there. Google signed me out of my notes. I'm flying blind here. Okay. I think the next thing we wanted to do was bring up another data layer here. So I'm going to zoom out a little bit, and I'm just going to hit the eyeball here. This hides the layer. So I can easily bring it up, or I can see what's going on by hovering over it. So if you have multiple layers, this is an easy way to clean things up so that you're looking at just one layer at a time. And I was going to show some biodiversity data off the Florida Keys. I had a tag selected. Okay, so this is the Florida Keys Reef Visual Census Biodiversity Indexing layer. So again, I can click on the title and I can go to a metadata page. This is going to give me all the metadata about how we visualize the data. You can find out how to download the data, and you can go back to the original data that we pulled from. But just going back into the map, or I can just hit the plus button. So now this data layer has been added to the map. And if I go back to the legend, you can see that it's loading. A quick way to zoom in on a data set is to just click on the magnifying glass. So now I can, this is something you can do with the sensor layer too. I didn't show you. You can click on the palette and change the color palette. You can change the opacity, and you can rescale the data. With this biodiversity layer right now, we're visualizing just number of measurements, but we can also look at biodiversity measurements like richness. Filter on species group classification, so we can look at just groupers. Or you can do a text search. This would be a way to, like, if there's a particular station code or something in the data that you want to find, you could use that. Or you can go under advanced and look at a full taxonomic tree, habitat, subregion, etc. You can filter on time using the time slider at the bottom. So this is a way to sort of get an animation over time. As I'm dragging the time slider, it's updating. Or I can arrow through it using the calendar. So now I'm advancing about 2 years each time. And again, I could click on the calendar itself and set up my start and end times to be yearly, just a full-time series, etc. If I'm interested in pulling a time series for this data, I can draw a polygon. So I clicked on the polygon in the upper left. So now we get a time series of richness data. The dropdown at the top lets me look at all the other visualizations that we offer for this layer. So I can look at the different, average length of species, and I can go through these. Say I'm interested in comparing this data to sea surface temperature. I can go back to find data, clear out my search, and I'm just going to search specifically for the GRIST layer. But another way to do this would be to click on tags and click on models here, models and forecasts. And this and other forecast layers would come up, or model and forecast layers. So if I click on sea surface temperature, then I get this model layer that comes up. And so now we're looking at, I'm going to just make my legend a little cleaner. We're looking at the Florida Keys biodiversity data, and it's overlaying sea surface temperature. So the time period that I'm looking at for sea surface temperature is October 2nd. But the biodiversity data offers data from 1994 to December 2nd, 2016. So I'm going to drag my time slider back to make a better comparison. So now we're looking at a time period in 2015 for the water temperature layer. And let's say we want to just look at data from the summer of 2016. I can pull my time slider in. So now we're looking at the spring and summer of 2016. So this is a better comparison. I can click on the legend of the sea surface temperature layer, pick a better palette. And I can also auto scale. The data looks pretty flat right now. So if I say auto set for data in view, it'll automatically rescale the data for so that the min and max are a little cleaner. If I hover over the water temperature layer, I get a preview of the data during the time period that I'm looking at. So right now it's looking at a time period in 2016, and the red is the exact time slice that we're looking at. If I click on the data, I can get a time series. This one takes a second. It's pulling, this one's pulling close to 30 years of data, I think. So this is a time series of water temperature, the gridded product for water temperature, 20 years of data at that spot. I can change the binning. Since we have more than 3 years of data, I can also create an anomaly plot. So there's a lot you can do here and explore. You can also download this data. In this case, we offer a CSV. And I wanted to use this as sort of an entry into another function that we have. And I referred to this earlier. This is called data views. So data views, if you see the blue star, the star up here, think data views. So any plot you're looking at, you should have a little star next to it, and you can add that plot to a data view. So when you click on the star, you see default and 0. So that's your default data view, and you can start adding data to it. So I can say save to data view. Then I'm given the option to add this to a comparison chart, and I'll say yes. So at this point, I have one chart in my data view, and my comparison chart is just that plot. And on the right side, it's just that plot. And then you can see this little 1 is sitting next to the plot on the right, and that's where we pulled that data from. So now I'll go back to the map, and let's compare this to some biodiversity data. So since we have 20 years of data, I'm going to expand out the biodiversity data to show the full time series so that we can look at this over time next to the water temperature data. So I'm just sort of randomly picking a region. So now we have another time series, and again, there's a blue star. I can add this to my data view and add it to the comparison chart. So now I have a comparison chart with 2 plots on it. And then on the right side, I have the individual plots. I can zoom in by dragging over the plot. And then there are some controls below for controlling the binning. So right now we're looking at yearly bins with the richness data, but let's make that monthly to compare directly to the water temperature data. And then, I can clean up the water temperature a little bit by removing the seasonal statistics and the min max. So now we're looking at mean temperature next to richness. And you can zoom in and, hover and get actual information about what's going on. If this were a data view that I wanted to share with folks, if I said, okay, I found something interesting, I want you to look at it and download the data and do something else with it, I can hit the share link and send it out. I don't see the chat. I'm going to send it to Lauren. Maybe you can add it. This isn't associated with a user account. So this data view only right now exists for me. It's tied to a cookie on my computer. By sharing the data view with everybody else, I'm basically sharing that cookie. It's now your data view to work with, any changes you make, I won't see and other people won't see. The default data views that I referred to earlier, these are some hurricane demonstrations. These are added to the portal and they are available for everybody. But again, those can't be changed by any user, at least for everybody else. You can change it for yourself and zoom in on plots and stuff like that. Okay, so I think next we were going to look at some of the shell-based data from within Georgia. And I am going to clear out some of these data layers. I still have the sensor layer open in case we need it. Okay, so I'm interested in shell-based data. I'm probably not the best person to explain what the shell-based data is, if Lauren or Abby wanted to jump in. But otherwise, you can look at the metadata and learn all about it.
Yes, this was a data aggregation effort to bring in water quality data in the SECOORA region, so in each of the SECOORA states. And so you'll see here that it's broken down by state, or you can get all 5 at one time. But it's mainly water quality data for shellfish aquaculture work.
Thanks, Lauren. And I did share a link. We just posted a story on it and did a webinar last Tuesday. I haven't had a chance to post it, but that will be a part of the shell-based story I shared.
Okay, so this works similar to the sensor layer and the biodiversity layer. As we zoomed in, we got dots and we can click on the dots and get data. So now we're looking at water temperature over time. If you wanted to see a different chart type, you could change it to line. Sometimes it makes it a little easier to compare. And you can also look at all the other variables available at this site. So right now we're looking at water temperature, but there's also fecal coliform, salinity, conductivity, dissolved oxygen, and acidity. So we could click on that again. This is missing my data view. I was going to make a data view here. For some reason, the data view function is missing here. I made a promise that wasn't the case. Well, immediately after the call, I'm going to have a look and see if I can fix that. But the idea would be that we could compare, fecal coliform to water temperature. I'm going to turn on the sensor layer and just kind of show you that we could do the same thing with sensors. So you could create a new data view here, manage data views, hit the plus. So we could say water quality comparison. Add to data view, add to compare chart, and then I can find a different sensor station. This is water level, so it's not really water quality. Let's see if we can get something better. This has water temperature. And I can compare this. Something that the shell-based data offers that the sensor layer doesn't is the Polygon tool. So if I'm interested in aggregating all the data from this region, I can draw a polygon around it. Oops, expand that out. Oh, and we did get our data view button back. So unfortunately, we can make a comparison. So it looks like it was just a bug with the feature selection. But now we're looking at an aggregation of water quality data from this area. So I can compare this. So let's add the fecal coliform and add this to our water quality comparison. So now we have a data view. In the upper left, you see the polygon that we selected, and then there's a 1 and a 2 referring to the 2 different sensor stations. The fecal coliform data that we looked at only goes out to 2021, so I'm going to zoom in on that time period. And those are the purple lines. So I'm just going to zoom in on the area that appears to have all of the different data types. So now we're looking at water level. I'm going to go ahead and hide that. We're looking at water temperature from the co-op station at Fort Pulaski compared with this polygon selection of fecal coliform data. And again, I can hover, get information, zoom in, change my time bin, etc. And I can share this out. One other thing I can do here that can be useful is click on the gear I can change my data view label, and I can also add a description. And that sticks with it. If I share this data view, you'll see the description, or I can click on this little pencil icon and I can, add an annotation here, check out. And I don't actually have any annotation, but that again, we'll stick with it. And then I can share this by clicking on the share button. Downloads are available here. So if I click on downloads, I'm going to get that fecal coliform data, the sensor station data, etc. I think that's all I had planned, and I think we're almost out of time, but if there are any questions or any data that anybody is particularly interested in looking at, I'm happy to go over that. I don't actually see any hands up, so somebody might have to speak up.
I did have one question come in. Can you hear me okay? Oh. So it says, how do you get this data information / how do you submit data to SECOORA?
Right. Sorry, I can't hear you very well, Brittany. But I did find okay.
So Brian, she's asking, how do you submit data to SECOORA? So if you have a data station, how do you get it in this portal?
Lauren, can you answer that?
Yeah. So generally, this is the data that gets into SECOORA is either funded by SECOORA or a SECOORA funded PI in the region. And it would be, if you are neither of those things, you would want to contact SECOORA and see if this was something that would be of interest to integrate into the portal. But for the majority of this data, it's data that's funded by and or of relevance to the SECOORA region.
Yeah, and I will say it includes it includes our SECOORA members as well. That's a perk of being a member of SECOORA, is we'll help you with the data management and get it in this portal so you have a record of it and the ability to showcase the data that you are collecting.
Yeah, and I'll say if you are a SECOORA member and you are submitting data to SECOORA through Axiom, we have a lot of staff on hand to help with various things. Visualizing your data, writing metadata for your data set and getting it public. We can also submit data to National Archives. So a lot of grants these days have requirements that your data go to a National Archive, and we have the ability to kind of streamline that process for you and make it a little easier.
Thanks so much, Lauren.
We had one more question. I think this one was for Rachel. Can you guys hear me okay? Okay, it says, does Sapelo ever coordinate with other NEARs in the Southeast through the SWMP program to compare data or create new projects, or do you just have the data available for comparison?
Yeah, there have been projects where we've done what they call system-wide comparisons. If you go into our NEARS Science Collaborative website, you can find some of those there. But, obviously, since we're constantly updating our data, there's room for more of that to happen. Thank you for including that.
Yes, the link is for the NEARS Science Collaborative is now in the chat. Are there any more questions? Anyone have any more questions?
So I did see a question come in. I'm not seeing on the chat though, but it came in through the Google form that you guys all filled out to register. Rachel, the permissions and use of the data, do they need permission to use it, or is it free to use?
It's free to use. That data policy that we have tells you, kind of the limits of the use and the limits of our liability for how you decide to use it. But it is public data that you can use. Again, we just ask that that you cite the data and you cite the reserve that the data came from.
Thanks, Rachel.
Okay, if there anybody else have any more questions? If you want to come off mute, you can to ask your question. Nope. All right. Well, thank you everybody for this great presentation and for showing us all the great ways that we can access virtual data in Georgia. I'm going to put in the chat if all the participants today could fill out this evaluation form and let us know how we did, if there's anything else that we could do in the future that would be beneficial to you, and how it went for you. Abby, anything else that you'd like to say or cover?
No, I just want to thank you guys so much for attending. And as Brittany said, there will be a recording. We'll post it on the SECOORA website as well, so folks have access to it. And please feel free to email us. I'm going to put our data management email into the chat, dmac@secoora.org. If you guys have any questions about either the NERS data or our data portal, please reach out to us. We're standing by and happy to help.
I will put my email as well in the chat too.
Thanks, Brittany.
Just Brittany. Okay. Well, thank you all for joining, and we'll see you on the next one.
Thanks so much.
everybody.
Have a good one. Bye.